Hello everyone, welcome. In this video we're checking out February-March 2017 variant 2. Before we get started, make sure to check out the notes that I have available for you in the description for chapter 1 of A-Level Bio. And if you have any questions, please make sure to write them down in the comments. Now let's get started with question 1. The diagram shows the outer structure of a typical animal cell, which structure synthesizes and transports lipids. That will be the smooth endoplasmic reticulum. C. Okay. A light microscope is used to observe two structures that are 200 nanometers apart. How far apart are the structures when the magnification is changed from times 40 to times 400? If they are 200 nanometers apart, they will always be 200 nanometers apart, no matter how closer you look. And so the distance between them is 200 nanometers. Question 3 says, the diagram shows the stage micrometer scale viewed through an eyepiece containing a graticle. The small divisions of the stage micrometer are 0.1 millimeters. This is the stage micrometer. And the small divisions are 0.01 millimeters. And then he says the stage micrometer scale is replaced by a slide of a plant cell. What is the length of the nucleus of the plant cell? Okay, so our goal here is to find the length of the nucleus. So let's see how many divisions this is. It's like 8 approximately from here, 65 I would say, until 75. So it's from 65 to 75, that's 10 units, right? So we need to find how long the units are in order for us to find the length of the nucleus. And so here we have the stage micrometer for this. 0.1 millimeters of stage micrometer is equivalent of 50 minus 10 units of the eyepiece graticle, and therefore this would be 40 units, and 40 units are equal to 0.1 millimeters. How many are 10 units equal to? And so we cross multiply, and we get 10 times 0.1, we divide by 40, and that should be 0.025 millimeters. Let's see our options. We have 0.8 millimeters, 8 micrometers, 25 micrometers, and 200 micrometers. So if we change to micrometer times 1000 to convert to micrometer, that will give you 25 micrometers, which is C. Some features of cells are listed. Which features are found in both animal and prokaryotic cells? Okay, so both of them have cytoplasm, cell surface membrane, both have cell surface membrane, ribosomes are found in both. But the difference is that in animal cells, ribosomes are 80S and 70S, meanwhile in prokaryotic cells, they're only 70S. So it's 1, 2, and 3A. Which size of ribosome is found in chloroplast? That would be 70S. In which of these organelles is ATP synthesized? Golgi body, mitochondrion, chloroplast. Now, the Golgi body does not synthesize ATP. However, my chloroplast and mitochondrion do, so that's D. A sample of milk is tested with Bendix solution. After boiling, a yellow color is observed. Which conclusion is correct? A high concentration of glucose is present. Actually, if you have a high concentration of glucose, you would get brick red, not a yellow color. Yellow means mid to low concentrations of reducing sugar. And you don't really know if it's glucose or if it's any other kind of reducing sugar. So this answer is wrong. A low concentration of sucrose is present. Sucrose is not reducing, and so testing for it using Benedict would not give you any result, would give you a negative result. You'll get blue and you're not going to get yellow. So this is also wrong. No reducing sugars are present. That is not what we can see here. If no reducing sugars were present, we would get blue. So cancel. Reducing sugars are present. That's the answer. The table shows some information about carbohydrate polymers. Which row describes amylose? Okay, amylose is supposed to have 1,4 alpha-glycosidic bonds and 1,6 amylopectin will have alpha-1,6 glycosidic bonds because it's branched. However, amylose is not branched, and therefore this is right. Shape of the molecule is helical, that is true, and therefore the answer is B. Which row about alpha-glucose and beta-glucose molecules is correct? Carbon atom on which the OH is present. So for alpha-glucose and for both alpha-glucose and beta-glucose, they're going to have OH present on 1 and on 4, but cellulose molecules contain both. Okay, cellulose does not contain both. So the answer should be no. But it's either 1 or 4. Oh, on which the OH position is different. Oh, okay, sorry. Okay, so since they're asking for the place from which it's different, then let's go ahead and draw alpha-glucose and beta-glucose. So So for alpha glucose, it will be down, up, down, 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 up, down. Meanwhile here in beta glucose, this is alpha, this is beta. It will be like this. It will be up, down, up, down. So you can see it's different over here, and that's it. Okay, it's going to be different on the what's it called, carbon molecules. This is 1, 2, 3, 4. So it will be different on 1. A. Some of the molecules found in animal tissues are grouped into 3 lists. Which list includes 1 or more molecules that contain nitrogen atoms? Okay, 1 or more. Let's see, glycogen, cholesterol, triglycerides, and water. Okay, cholesterol and glucose obviously does not contain nitrogen. Water does not contain nitrogen. Cholesterol and triglycerides do not contain nitrogen. Cholesterol, however, it's also a lipid, so I'm not really sure if it will contain nitrogen. But let's see 2. Glycogen, antibodies, adenine, phospholipids. Okay, here you can see it's definitely one of the cases because antibodies are proteins. Proteins do have nitrogen because they have amine groups and everything. Adenine is nitrogenous base, and so from the name we know it has nitrogen. Phospholipids do not have nitrogen. Neither does glycogen, but we have, as you can see, 1 or more molecule that contains nitrogen. So we could choose 2. Hemoglobin, carbon dioxide, mRNA, and monosaccharides. Hemoglobin and mRNA contain nitrogen because hemoglobin is a protein and mRNA is actually formed of nucleotides which have nitrogenous bases, and therefore 3 and 2 only. Okay, now for question 11, hemoglobin consists of 2 alpha chains and 2 beta chains. Approximately 5% of all humans have 1 amino acid in the beta chain that has been changed, affecting the structure and stability of hemoglobin. Which levels of protein structure could be changed in hemoglobin of these humans? Obviously, primary will be changed, and if primary is changed, then that could change tertiary structure, and that also could change secondary, and hence could maybe change quaternary. But but obviously, like here, he says that all humans have 1 amino acid in the beta chain. It has been changed. And so what does this change? It will change the primary structure. It will obviously change the tertiary structure, but we don't have an option for primary and tertiary only, so we must pick primary, secondary, tertiary, and quaternary. C. Which row gives the correct description of both a collagen molecule and a collagen fiber? Okay, a collagen molecule, alpha and beta polypeptide chains forming a double helix, held together by disulfide bonds. A polypeptide chain with repeating amino acids forming an alpha helix. Okay, definitely not. There's no alpha helix in collagen. A polypeptide chain with 3 repeating amino acids forming a helix. 3 helical polypeptide chains forming a triple helix held together by hydrogen bonds. This is true. Collagen fiber, however, is molecules of collagen lying parallel and cross-linked to each other. That is correct, so it's D. The diagram shows the structure of 2 amino acids, each of which has 2 amine groups. A peptide bond is formed between 2 amino acids. Which groups could form the peptide bond? Okay, which groups could form a peptide bond? So here we look at CH and CH and... Okay, so obviously here the peptide bond is going to be formed between 2 and 4. It also could be formed between 1 and 5. So let's see the options. We have 1 and 4. 1 and 4, no, obviously not. 1 and 5, no. No, actually, that's right, 1 and 5. So B is their answer. 2 and 6, absolutely. No, 6 you could see is actually part of the R group. Okay, it's not the, it's not going to be part of a amine group, you could see here. And then 2 and 6, 2, as we mentioned, 2 and 6 is wrong. 3 and 5 is also wrong because this is also the R group. Okay. An enzyme was added to a small excess of its substrate. All variables were kept constant. A student was asked to sketch a graph to show how the concentration of the enzyme substrate complex changes over time. Which graph shows this correctly? I would say that since the enzyme is added to a small excess, then that would mean that the concentration would be constant for a specific period of time until it starts to decrease when the substrate starts to decrease because, it's used up and it's turning into product. And so I would say the answer would be D. Okay, the table contains results recorded by a student from an investigation to the effect of temperature on an enzyme catalyzed reaction. All other variables were standardized. What's the correct conclusion? 40 degrees Celsius was the optimum temperature. Okay, let's see the temperatures and look. Okay, so temperatures here you could see as temperature increases, the rate of reaction increases all the way until 50 degrees Celsius, where it starts to decrease from here on. So what's it called? We could say that okay, so let's see what are he says here 50 degrees Celsius was the optimum temperature. I don't think so. I mean, we have a huge gap between the temperatures. There's like a whole lot of like 10 degrees between us, and so it could be, for example, 38, it could be 35, not necessarily 40, and so I would not say it's 40. The data for 50 degrees Celsius was anomalous, not necessarily. I mean, it seems pretty accurate to me. The optimum temperature was between 30 and 50, or the optimum temperature was between 40 and 50. Okay, so between 40 and 50, the rate decreases, it doesn't really increase. However, between 30 and 40, it kind of does decrease. And so, the optimum temperature is anywhere between these guys, because we don't know, for example, if it goes to 34, then it decreases, or if here it was at 34, then it decreased. And so it could be anywhere between 30 and 50. Don't narrow your window of options, and so C should be your answer. 17. In the cells in the roots of beetroot plants contain red pigment. When the pieces of the root tissues are soaked in cold water, some of the red pigment leaks out of the cell and into the water. An experiment was carried out to investigate the effect of temperature on the loss of red pigment from the root cells. It was found that the higher the temperature of the water, the higher the rate of loss of pigment from the root cells. Which of these statements could explain the trend? Enzymes in the cells denature as the temperature increases, so the pigment can no longer be used for reactions inside the cells and diffuses out. I would assume that if the pigment was to be used for reactions before, we wouldn't have actually seen it. It would have already been used up. But no, it's not really used for reactions. It's just there for because the plant looks this way. As the temperature increases, the tertiary structure of protein molecules in the cell surface membrane changes, increasing the partial permeability of the membrane. That is true. When the proteins change, when the temperature increases, that could denature the protein molecules and change their shape, and so as a result maybe increase the permeability of the plasma membrane. And I think 3 here says phospholipid molecules gain kinetic energy as the temperature rises, increasing the fluidity of the phospholipid bilayer and allowing pigment molecules to diffuse out more easily. Same thing. Phospholipid molecules, when we increase temperature, the phospholipid bilayer becomes more fluid and more permeable to more things, and therefore 2 and 3 are my answers, and that should be B. Okay, the cell surface membrane structure is described as a fluid mosaic. What statements describe the mosaic part of the cell surface membrane? So the fluid mosaic is descriptions is given to the phospholipid bilayer as a result of the phospholipid heads and the protein molecules moving around, and from the top view would look like a mosaic type of structure. Different patterns are obtained by the moving phospholipid molecules. It's not just the phospholipid molecules. The random distribution of cholesterol molecules within the phospholipid bilayer cholesterol has nothing to do with it. The regular pattern produced by phospholipid heads and membrane proteins, I don't think that it's regular, really. It's it keeps on changing. The scattering of different proteins within the phospholipid bilayer, I think so. The proteins will be scattering around in the phospholipid bilayer, and the top of them is going to show as like it's moving around alongside the phospholipid heads, and so that's D. The diagram shows 2 pathways X and Y, which molecules can diffuse across a cell surface membrane. Which row correctly shows possible pathways for lipids, water, and glucose? Obviously, lipids, cannot pass, through, these guys, the protein structures. You could see obviously here, if we need a channel protein, then we could be water, we could be glucose, but X could also be a pathway for water. However, lipids can only pass through the phospholipid bilayer, and so I would say lipids are going to be X only. Okay, water is going to be both X and Y, and then glucose is going to be Y only. A. Some stem cells divide and rise to phagocytes some stem cells divide and give rise to phagocytes. Where in the human body do these stem cells divide? They divide in the bone marrow, and that would be it. Okay, so remember that cells that lead to blood cells, in this case white blood cells, they will be dividing and differentiating in the bone marrow. Okay. What is the correct sequence of stages in the mitotic cell cycle? So we start with G1, and then we move on to S phase, and then G2, and then mitosis, and then cytokinesis, and that would be C. So let's remember together how the cycle looks. We're going to have what's it called, let's say mitosis between this section and this section. And then before mitosis, we're going to have G2 phase, which will last quite a long time. We're going to have S phase over here. Okay, so we have G1, first growth period, S phase. And then we have G2, and then we have mitosis, which is split into 4 sections. Okay, so here, oh my god. Okay, so this would be G1. This is the first stage, stage 1. And then here we're going to have the synthesis phase in which we're going to be synthesizing what's it called, our proteins, our kinetochores, which are going to be what's it called, part of your chromosome, replicating DNA as well. And then what's it called, we're going to have G2 phase, second growth period. That will be the phase in which we will correct DNA for any mistakes, if there were any. Also make sure to replicate centrosomes before we start mitosis, which is going to be from here until there. This is mitosis. And here we go P mat. P for prophase, M for metaphase, A for anaphase, and T for telophase. Okay, let's move on. Then we have what's it called, the enzyme telomerase repairs telomeres. It stops telomeres from getting shorter each time a chromosome is replicated. Telomerase is not normally active in human body cells, but in some diseases telomerase can be activated. In which disease is the enzyme telomerase activated? Is it cancer, HIV or AIDS, malaria, or myasthenia gravis? So I would say that's cancer, because why would cancer cells need telomerase? Telomerase allows cells to replicate indefinitely. Telomerase can maintain telomeres, and telomeres determine the lifespan of the cell. Basically, telomeres are the ends of the chromosome that do not code for anything, and they're just there for as a protective mechanism, because the enzyme DNA polymerase doesn't run until the end of the DNA molecule, and so what's it called, the DNA strands keep getting shorter every time the cells replicate, and so this will inevitably result in the death of the cells. So in order to make sure that we are able to replicate indefinitely, we have the enzyme telomerase, which keeps on, making the DNA molecule longer and longer and maintaining the telomere, so that whenever DNA polymerase thinks that it's so smart that you remove part of your DNA, no, it actually remove part of your telomeres, which doesn't really code for anything, so we don't actually need it. 23. The statements describe events during the mitotic cell cycle. What is the correct order of the 3 events in the mitotic cell cycle? So you have 1, chromosomes migrate to opposite poles of the spindle. 2, chromosomes arrange themselves at the equator of the spindle. Chromosomes condense and the nuclear membrane disappears. Centromeres divide. Okay, so the first thing that happens, so this will be part of anaphase. Anaphase is the 3rd step. Okay, and what's it called? Chromosomes condense and nuclear membrane disappears. It's part of prophase, which is the 1st step. And then we have chromosomes arrange themselves at the equator. That's part of metaphase, M for middle, and that's the 2nd step. And then lastly, we have chromosomes migrate to opposite poles of the spindle. Okay, the chromosomes migrating to opposite poles will be part of the end of anaphase, beginning of telophase. So I could just write here telophase, as in it's the last step. That would be 4th. It goes P-M-A-T, P-mat. Okay, prophase, metaphase, anaphase, then telophase. So here, what's it called? The first should be 3, then 2, and then 4, and then 1. So 3, 2, 4, then 1. So that's B. Okay, which statements about nitrogenase bases is correct? Okay, nitrogenase bases. Adenine is a pyrimidine. No, actually, adenine is a purine, and purines have double ring structures, so the entire statement is wrong. Cytosine is a purine with double ring structure. No, cytosine is a pyrimidine. Guanine is a purine with a single ring structure. If guanine is a purine, it should have a double ring structure. Uracil is a pyrimidine with a single ring structure. That is true. Rifampicin is an antibiotic used to treat tuberculosis. It works by inhibiting RNA polymerase in bacteria. Which of these processes will be directly inhibited by this antibiotic? ATP synthesis will not be directly inhibited because it has really nothing to do with transcription. ATP synthesis is part of, what's called aerobic respiration, mitochondria, maybe it's part of photosynthesis, but not part of transcription and protein synthesis, so we can cancel it. Transcription will be directly affected. Translation, however, not really directly affected, and there is no option for 2 and 3, so we'll only pick 2. Okay, the first generation is, let's imagine how the experiment will go. So basically we have a medium which basically contains heavy nitrogen. And we have a bunch of cells in it, okay? So we have cells here. How does the DNA of these cells look? It looks something like this. We have, what's it called, 2 DNA molecules as part of the chromosomes, and they're going to, be basically entirely made of heavy nitrogen. Now take one of these cells and put it in another medium. Okay, let's imagine it's something like this. This is light nitrogen medium. Okay, and we put here a cell that had heavy nitrogen DNA, so the 2 chromosomes. Now we're going to have it replicate inside the light nitrogen medium, and so it's going to produce a cell that has slightly different DNA because DNA replication is semi-conservative, and so we know that's going to be something like this, right? Because what happens is we have obviously 2 cells when it replicates, so one will be this color, the other one will be blue, okay? How does this work? So basically, the DNA replication is going to happen as follows. Each one of these strands is going to be replicated. So one of them is going to be maintained, and the other one is going to be made out of the light nitrogen medium as a complementary copy. One of them will be maintained, the other one will be made out of as complementary from the light nitrogen. Now this is the first generation, and it's obviously hybrid because it has both heavy and light nitrogen, and so the first generation is entirely hybrid. Okay, but then the third generation, so now take what's it called, one of these guys, and let's have them replicate inside again light nitrogen. So what happens? This guy is going to be maintained, right? But what happens here is we have a copy which is made of light nitrogen. Then this guy is going to be maintained, and we have a copy of light nitrogen. So this is the second generation. Now take this is going to be our third generation. Okay, we have 2 different guys here, and okay, sorry, the color keeps changing. Now these guys are going to replicate, so we're going to maintain this, right, and we're going to make a copy, and then we're going to maintain the blue and make a copy using light nitrogen again, and then we're going to maintain this and make a copy using light nitrogen, and then maintain this and make a copy using light nitrogen. And so you could see what happens here. We have only 1 hybrid and 3 purely made out of light nitrogen, and so it's a 3-to-1 ratio. So what do we have here? Half hybrid, half light of the third generation. No, the third generation is 1/4 hybrid, 3/4 light, so that would be B. The diagram shows stages in the production of part of polypeptide, DNA nucleotide sequence, template strand, okay, and then we have mRNA sequence, and then we have the amino acid sequence. Which feature of the triplet code is illustrated by the information given? So an amino acid coded for by more than 1 triplet. Yes, so we could see that here. We have these 2 amino acids, and we have this triplet code and this triplet code. So obviously, it's coded by more than 1 triplet. So this is true. The triplet code is universal for DNA of all organisms. Again, this is not what's represented in this diagram here, because if we were to say, for example, have a prokaryotic cells DNA nucleotide sequence template strand, and then have a humans, okay, and this would for example show us that they both have the same template strand, or same amino acids for the same codes. Sure, this would be represented here, but this is not what's illustrated by the information. There are some triplets that code for start and stop, again not shown here, so we could just immediately go A. Okay, so then we have which combination of features is characteristic of flume sieve tube element as it unloads into the sink. Okay, so as it unloads into the sink, we have water potential higher than sink, or lower than sink. It's going to be higher than sink because the material is going to flow within the flume sieve tube element by mass flow, so down water potential gradient. We have a high water potential in flume sieve tube compared to the sink. And then we have lignification of the cell wall is going to be absent, of course, it's not lignified. So it would be A. 29. Which statements correctly describe transport pathways in dicotyledonous plants? 1. In a symplast pathway, water may move through intercellular spaces. 2. The symplast pathway may be blocked by the tonoplast. 3. In the aplast pathway, water does not move through plasmodesmata. And 4. The aplast pathway may be blocked by the Casparian strip. Okay, so 4 is definitely true because the Casparian strip is found in the cell wall, and it is a strip that's made of what's it called, waterproof material, and therefore submarine waterproof material, which is going to obviously block the pathway of water. So the upper pathway, the dead pathway, is blocked. And then also 3 is true because Laplace pathway water does not move through plasma matter that is part of the Simplast pathway. Now tonoplast does not block the Simplast pathway, and also what's it called, in Simplast pathway intercellular spaces are dead. Okay, they're not really living parts of the cell plant, and therefore it's not really Simplast, it's not really alive. And so we can cross 1 and 2 and choose 3 and 4. The diagram shows transverse sections of parts of the plant. So we have what's it called, leaf and stem, which label structures transport mineral ions. Mineral ions are transported in the xylem. 1 is the xylem and 3 is the xylem. So 1 and 3, however 2 and 4 are wrong, so yes, A. Which feature of transport in plants is correct for both xylem and phloem? Okay, so it is passive. No, in the phloem it's kind of active. It occurs by mass flow. Yes, it does occur by mass flow in both xylem and phloem. It occurs from source to sink. That's not true for xylem. It occurs only in one direction. Again, that's not true for phloem. And therefore, what's it called? All of the options are wrong except for mass flow, B. What is the main function of the companion cell in the phloem tissue? Providing cytoplasmic contact with the sieve tube element for loading, providing structural support for the sieve tube element, providing the nucleus for the cell division in the phloem, providing the source for assimilates for storage. Okay, so obviously it's not going to be there to support the sieve tube element, and it's not there for cell division, and that leaves us with providing cytoplasmic contact to the sieve tube element for loading, which is exactly what it does, and so it's A. The contraction of the heart is coordinated through electrical impulses passing through the cardiac muscle. What is the correct order for of sequences of these impulses? Okay, so we have right and left atria. Okay, so right and left atria to the atrioventricular node, to the Purkinje tissue, to the ventricular walls, or right and left atria to the Purkinje tissue. Okay, so the sequence of the impulse will start from the sinoatrial node, which will pass it to the right and left atria, and then from the right and left atria, that's going to move, to the atrioventricular node, and from there to the Purkinje tissue. And so I'm not really convinced with option letter D because of the way that's written. However, option A does show us this, but without the sinoatrial node, and so A is our answer. It goes from sinoatrial node to right and left atria to atrioventricular node to Purkinje tissue to ventricular walls, and so this is true. As blood flows from an artery to a vein, the thickness of the walls of vessels changes. Which bar shows these changes correctly? So the thickness of the walls of the vessels. Obviously, arteries have the thickest walls, after which we have, I suppose that this is arteriole, capillary, veinole, and vein. Okay, so obviously capillary will have the least thickness, and then I would suppose it's going to be D, because it shows that our capillary have the least, artery have the most, and vein will have less than the artery and also less than the arteriole. Okay, next we have 35. The graph shows the oxygen dissociation curves of hemoglobin from 2 species of mammals, S and T. So you have partial pressure of oxygen, percentage saturation of hemoglobin with oxygen, and which statements could explain the difference in the oxygen dissociation curves of species S and species T. Species T has a lower hemoglobin concentration in its red blood cells than species S. I would not assume so. I would assume maybe they have, since they have the same, here, hold on, let's read all the other options. The hemoglobin in species T has lower affinity for oxygen than hemoglobin in species S. That's what I was going to say. T has a lower affinity than species S. Species T lives at higher altitude than species S. If you live at higher altitudes, that would just mean that maybe you have more red blood cells, but this will not affect your hemoglobin's, percentage saturation, because overall we're testing for the same partial pressures of oxygen. We're not really depriving T of oxygen. And so I would cancel out 3. And option 1, species T having lower hemoglobin concentrations, I would not assume so, and so 1 is wrong, and 2 would be my only answer, C. Which tissues may be found in the bronchioles? Okay, so bronchioles, do they have cartilage? Actually, they don't have cartilage. Okay, so cartilage-shaped epithelium, they do have shaped epithelium, but don't have cartilage. And then we have, smooth muscle, they do have smooth muscle. So I would say my answer would be, we could cancel out all, what is this, Benson? Okay, we could cancel out these three options and we'll remain with B. The effect of carbon monoxide in a cigarette smoke. Okay. It binds irreversibly to hemoglobin. So obviously smoking is actually out of your syllabus by now, so we don't really need to solve this. But one of the things that you might want to know is that carbon monoxide actually, causes it binds irreversibly to hemoglobin. That's true, and it causes mucus to does not really cause mucus to accumulate. It results in more carbon dioxide being transported in the blood. It temporarily increases the heart rate. I would say it's 1 and one only. Yes, okay. So it's B. And the global mortality figures for some diseases in 2002 are shown in the table. How many millions of people died from viral diseases listed in 2002? Okay, viral diseases here include HIV and measles. Malaria is caused by fungus and tuberculosis is caused by bacteria. And therefore, how many people died, not percentage of all this, how many people died. So I would add 2.8 to 0.6. 2.8 plus 0.6 is supposed to be, let's add. Okay. And this will be, wait, am I right? 3.4 is not, 3.4, B. Okay, you could tell I'm not really great at mental math. Anyway. Which of the statements could describe both B lymphocytes and T lymphocytes? They contain specific protein receptors in the cell's membrane. Both of them do, that's right. They differentiate into plasma cells. Actually, no, that's a function of B lymphocytes only. They divide by mitosis, true, of course, so 1 and 3. That's A. Two people, G and H, were each given an injection to protect them against a particular pathogen. One person was injected with antibodies, the other person was injected with a vaccine. The graph shows the concentrations of the antibody against this pathogen in the blood of the two people, G and H, over a period of 20 days following the injection. Okay, so we have antibody concentration from time of injection and over time of days. So you could see here that so one person was injected with antibodies, the other one was injected with a vaccine. So obviously here you could see antibody concentration for G decreases over time, meanwhile H, it's going to increase later on. So which row describes types of immunity shown by G and H? So G shows artificial passive immunity, meanwhile H will show it will also be artificial active, because it's not really that they had gained a disease, they were injected with a vaccine. And so I would say artificial active immunity and artificial passive immunity for G and H, that's going to be B. So that's it for this variant. I hope this video was useful. Thank you guys so much for watching. Make sure to like, subscribe, and share this video with anyone who might need it. Again, see you later. Bye.